Welcome to Chapter 16. This is mini lecture 2. But we're going to pick up right where we left off, talking about organisms and how they can become a pest. So now let's look at microscopic organisms. We'll look at mycoplasma and phytoplasmas. First off, these are very small organisms. They're very similar to bacteria. One of the main differences between bacteria and these organisms, these microorganisms, are that they have both the RNA and the DNA, but they lack a true cell wall. So we can't really grow it in a lab because it has, or on artificial media, growing media because of that. So that's really hard to detect sometimes due to the way we have really looked at. So that's one of the big differences between these small-celled organisms and bacteria. And then this is transmitted by insects, so it can't be really anything else, obviously, because it's an organism. What happens is a lot of the color changing of leaves will start to appear, and then stunting will happen. There's a decline in the plant really quick, unfortunately, a lot of times. And the Ester yellow disease is really one of the common ways that we see these organisms really take over plants. All right, nematodes. Nematodes, when you take soil, you'll learn a lot more about nematodes. We'll just go over a little bit of it here in crops, but they're microscopic and they're a roundworm. So I'll show you a picture of them in just a second, but they feed on the stylet of that plant and will do this extremely quick, and so it'll cause some chlorosis, some stunting, and some wilting in all of the plants very quick, and so one way that we stop that from happening, again, and you'll hear me say this a lot, is crop rotation. So if we have a nematode present in wheat, we probably won't take that, we won't grow wheat in that field the next fall. We will rotate that crop out probably until we are sure that something with our soil is not going wrong. So root knot nematodes are very common, and then the soybean cyst nematode causes a lot of problems, unfortunately, with the soybean crop in the United States. All right, so those are pictures of, on the left, there's the soybean cyst nematode, and then there's the egg. So you can see it kind of looks just like a roundworm that would happen in livestock or sometimes humans, unfortunately. So, and there's the eggs that you can see, and just like in any sort of mammal or human or any one of us, could happen in plants too. So parasitic, there's some very common parasitic flowering plants that have a really bad time with these. This will really hinder the root systems within parasitic flowering plants, and it can stop chlorophyll. So if it stops chlorophyll, we know it's going to stop the photosynthesis, and then that plant's going to go downhill really quick. And they really have a tendency to use that plant host to absorb those water and minerals. So it's kind of like it is just stopping all of that water and the minerals to the root system, to the plant. So we manage this by preventions. You actually go in and remove it with your hand. Herbicides are a very common way. Planting resistant and tolerant varieties. Again, that's a huge science in plant and soil science is finding varieties that will be tolerant to parasitic flowering plants. The dodder, the witchweed are very good examples of those. All right, so now let's look at insects. So insects have a very unique way about them and how they interact with plants. So the first thing you have to know is they're invertebrate. They're small invertebrate animals. We'll go over some body parts of them in just a little bit, but they have three different types of life cycles: metamorphosis, incomplete metamorphosis, and then no metamorphosis. And I'm going to show you some good examples of these in class this week, and you're going to be able to identify them. You're going to be able to tell me the life cycle of them and their feeding habit and this will be specific to crop plants. So they can either feed by sucking, piercing, or chewing, and that's how we identify them. We identify them by the life cycle and their feeding habits. So we talked about the three life cycles: no metamorphosis, incomplete metamorphosis, and then complete metamorphosis. So you can see no metamorphosis is the most the most usual, so the most original, I guess, as you would say, goes from an egg to a juvenile to an adult, okay? But in incomplete metamorphosis, you have nymphs. So you have an egg, then you have 3 different stages of a nymph stage, then you have an adult. Incomplete metamorphosis, you have an egg, a larva, a pupil, and then an adult. So you go from no metamorphosis to incomplete metamorphosis. Now, how does that interact with crop plants? Well, it just kind of depends on what crop plant it is and what that insect would thrive on. So we'll talk more about that in class. All right, so here's the difference between a piercing insect and a chewing insect. So you can see by what we call the mandible, or what we would also call the mouth on the left-hand side, the chewing insect, that obviously is going to chew. But the piercing insect, very, very similar, but it's just going to pierce, and it's just going to more of a piercing eating type of way about it. So those are the mouth parts of these 2 types of insects. All right, so important insect pests that we see, very common around here, are aphids, especially soybean aphids, and we'll talk more about that. Weevils in pecan trees. We have corn earworm that we've seen a lot, and grasshoppers. Those are very, very, very detrimental to a lot of crops, more so being soybeans. So here's a soybean aphid on the left-hand side, very yellow in color. Very, very, very destructive to soybeans. And then the Colorado potato beetle, but they'll feed on more than just potato leaves. So now let's dig a little bit deeper into the IPM, or the Integrated Pest Management System. So all this is just a concept that pests are always present. So we want a plan for whenever there are pests present. So we want to be able to go to a producer and say, what is your IPM for an aphid? And that producer would be able to pull out a plan for that, for what he or she values, for what he or she would like to do, the money that they would like to spend. So that's kind of the whole point about it. Now, what's the objective? Just to keep those pest populations low. Unfortunately, I don't think we're ever going to be able to eradicate a whole pest population, but we can have a plan in place for that to happen. So 2 goals: has to be environmental friendly for most people, and it has to be very cost effective, unless you are a rich farmer, which I don't know many of those. So those 2 are very important. So 5 main ways we look at this in an ag sense: cultural, biological, mechanical, genetic, and chemical. Genetic and chemical are kind of 2 in the same because they work very hand in hand together. So the resistant varieties, the fungicides, anything that we can do as a science to help that out. Mechanical, so either if it's hand picking or tilling. Biological, figuring out what insects and bacteria are in the soil already, if there's any green manure that's in the soil, and what might be present in that. And then crop rotation, like we've talked about, fertigation, and sanitation, all of those very cultural aspects of IPMs. So, chemical pest management, we have many different types. We have antibiotics, we have nemicides, we have insecticides, we have fungicides, like we talked about. So there are a lot of different ways for a producer to pick how they want to treat that pest. All right, so this is a graph that'll show you the percent of area treated with insecticides, and then on the bottom there's a crop with the year data taken. Okay, so corn, 2005, only about 25% of the corn was treated in 2005. Peanuts, though, however, because they are so susceptible to so many pests, were treated a lot more than some of the other crops that we have in the United States.